I'll be back like that too.
Yeah.
It smells like 3 a.m. Like,
Yeah. I thought you just like that. Whatever we had, you have it.
Yeah, but I don't know.
There's glucose metabolism.
Okay, so today we are going to finish up our discussion of metabolism. Okay. So last time we were talking about glucose metabolism essentially. So we talked about how you can take a glucose molecule, our C6H12O6, add some oxygen for aerobic cellular respiration, and break the bonds in that glucose molecule through a series of steps to get us a lot of ATP. So we talked about glycolysis, giving us 2 ATP on net. Talked about how we end up with pyruvate that has to move into the mitochondria and create the Krebs cycle as a single A, giving us oxygen acetate for that cyclic cycle, giving us a little bit of ATP along the way, but critically, lots and lots of electron carriers, reduced electron carriers. We talked about the electron transport chain, how embedded proteins in that inner membrane of the mitochondria build up a proton gradient, so lots of positively charged particles in the intermembrane space, so the space between the 2 membranes of the mitochondria, and how we can then release that gradient to power energy, to power the oxidative phosphorylation of ATP when we let that big gradient run through ATP synthase, essentially swirling it around like a water wheel or windmill or something like that. So that was our aerobic cellular respiration. So it requires oxygen because oxygen is our final electron acceptor in the electron transport chain. And then we talked about what we can do in a low oxygen environment. So we've used up all our oxygen, or for some other organisms, if they just live in a low oxygen environment. We talked about the fact that there is an alternate process, an alternate sort of pathway we can do to make ATP, which is that we can run glycolysis over and over and over again to just get that 2 ATP, 2 ATP, 2 ATP. But we encounter a problem, which is that we start to run out of NAD+, which does happen to be turned into NADH during glycolysis. So to make up for that, to regenerate that NAD+, that's part of the process, we can turn pyruvate into lactate. So when people talk about lactic acid buildup in like a muscle or something, that's what they're talking about. They're talking about the fact that that muscle cell has used up all its oxygen or can't use oxygen in the first place. So it's just doing that glycolysis process over and over again in the cytoplasm, still gets energy that way, but there's kind of a cost there. Okay. So that's what we talked about on Monday. That's where we left off. So now we're going to be talking about how we can get energy from things that are not glucose. So glucose is our main form of energy in terms of what we are looking for in our diet, right? Carbohydrates are a real quick hit of energy. Glucose is basically sugar. But we can break down other types of molecules. So it's important to be aware that those exist. Okay. So carbohydrates, great for a quick hit of energy. But we actually have a longer sort of term storage in our body. There are lipids, like fat, stores energy essentially in the molecules. We also have a carbohydrate storage molecule that we'll talk about as our first thing, something called glycogen. We can also break down proteins. Your body kind of tries to go to those last. You don't want to start to break down your muscle cells and harvest those proteins if you have, some nice sugar in your belly, or you have some fat cells, some triglycerides that you can break down, but eventually you do break down proteins as well. To understand that process, we want to remember our, trying to see if I can make these pictures go away. There. We want to remember our overall aerobic cellular respiration, because we're going to be basically going into this workflow at different points. So we want to remember that we're going from glucose through glycolysis, making some ATP, and glycolysis. Our carbon ends up in pyruvate. Okay. Pyruvate turns into acetyl-CoA. The acetyl-CoA in the matrix of the mitochondria then goes through the Krebs cycle, also known as the TCA cycle. This then goes into the electron transport chain, and we do oxidative phosphorylation, where we need oxygen and make lots and lots of ATP. So this is still going to be crucial to how we process other types of molecules, because as you can see here, right, we're going to take our proteins and we're going to feed them in to just essentially some of the later stages of this process. So we're not coming up with a completely different system. We're kind of just entering at different points. And same thing for our triglycerides. So our fats and oils, one of our main forms of lipids. But before we talk about our lipids or our proteins, we're going to talk about glycogen. So glycogen is in animal cells. The analogous thing in plants is starch, and it's storage for glucose, essentially. So down at the bottom there, what we are looking at is a glycogen molecule. I think we had this as one of our examples on our carbohydrate slide when we talked about how to identify carbohydrates. But you can see those glucose monomers building into our larger polysaccharide. So our polymer glycogen is made up of a bunch of little glucose subunits. So they're just our building blocks. And we have this sort of branching chain form. We're just stringing them all together like beads. Okay. So glycogen is found in specific places in your body. You don't store glycogen everywhere. You specifically have glycogen in your liver and in your kidney. That's the places in your body where you're going to do this storage. Okay. So you can store glycogen in your liver and in your kidney, and you can wait until the blood is low on glucose and then start to break down this glycogen, release those little glucose monomers from this larger molecule and send them out. And then your brain, for example, can then break down its little glucose chunk to make energy in the form of ATP in our normal aerobic respiration way. The reason this happens in specific places has to do with the enzymes that are present in these different tissue types. Okay. So, the thing that is true is that when you break off one of the glucose little monomers, you don't immediately end up with glucose. That's one thing. You need an enzyme to turn into this broken off little piece into glucose that looks like normal glucose if you just eat glucose on its own. Okay. So only liver and kidney cells have the enzyme that you can use to do that. So glucose 6-phosphate is what you would end up with if you just broke right here. Okay. And so in order to make that normal glucose, you need to use the enzyme glucose 6-phosphatase. And then glucose is what would leave the cell. So we have a kind of stepwise process, right? We'd go from glycogen, we'd break off little bits of the glucose 6-phosphate. Okay, I'm being lazy because I draw slow with a mouse. So that's our step 2. Then we need our enzyme, that glucose 6-phosphatase, right? The ACE. Those of you who have prettier handwriting, I apologize. But your notes are beautiful. Okay. So that's our glucose 6-phosphatase, is an enzyme. And that's how we end up with actual glucose. So that middle step, that intermediate step, the glucose 6-phosphate here in the middle, that molecule can't get out of a cell on its own. It's just kind of stuck. So we're not saying you can break down glycogen, but there wouldn't really be much of a point in terms of fueling the rest of the body. We're going to look at a whole big chart of what's going on with glucose, that glucose 6-phosphatase, and the glycogen. But to understand the chart, we want to know one other fact first. Okay. So this other fact is that you can actually make glucose in your body. You don't have to eat glucose. I mean, long term, you should eat glucose. But in terms of small scale time, you can make new glucose if you don't happen to have any. So the word for this is gluconeogenesis. In general, in this next few slides, I just want you to read words really carefully. And when you get to the exam, read really carefully because there are a lot of words that start with a G. If you slow down, they do tell you what they mean. So gluco for glucose, neo for new, genesis for creation. So what this word is telling you is that we can create new glucose. Right? That's what we get if we read this kind of backwards. Gluconeogenesis. So gluconeogenesis is the process of synthesizing, making new glucose from other things. So we do need stuff to build the glucose out of. So our substrates for this would be things like glycerol, lactate, or amino acids. Okay? So we can build them out of other types of molecules. Right? Our glycerol was the glycerol backbone of a triglyceride, but on its own, it's kind of an alcohol. Lactate we saw was something we get from pyruvate when we're doing our glycolysis, anaerobic respiration, that sort of fermentation process. Another way to talk about that. And our amino acids were our building blocks of proteins. So we can take other classes of molecules and make new glucose out of them. This mostly happens in your liver. So your liver is really important where we're thinking about control of your glucose because it's storing glucose in the form of glycogen, and it can also make new glucose if it needs to, right? And this is not exactly like just running our cellular aerobic metabolism backwards. It's not even quite like just running glycolysis backwards. So you don't exactly build up glucose the same way you build it down. You're going to need some specialized enzymes in order to do this, which again is why it's going to occur in a specific tissue type, a tissue that has those enzymes. So in order to make our glucose, we're going to take glucose 6-phosphate plus glucose 6-phosphatase. We could make glucose when we're breaking down our glycogen. The other thing that we can do is use something called hexokinase. So we're gonna look at that on the chart on the next slide because I think that's actually clearer how it works. Okay. So overall, the many terms that we're trying to make sure that we can tell the difference between, right? Glycogen was our storage molecule. So our big polysaccharide, our carbohydrate that we mostly have in the liver, the kidney. We had our gluconeogenesis, which was how we build up glucose from other things. We make new glucose. We can also have our glycogenolysis. So here you want to pull out the glycogen for glycogen and lysis for breaking down. And here is glycogenesis. So this is glycogen genesis, essentially, so making glycogen. Okay. So what happens here? I'll erase those highlights. Okay. So if you're starting with a glycogen molecule in the liver, when you're breaking it down, you're going to circle glycogenolysis, lysis for breaking, glycogen for glycogen. When you break down glycogen, you get glucose 6-phosphate. So if we want to turn like a glucose 6-phosphate into glucose. We then need our enzyme, glucose 6-phosphatase. So here, essentially we broke down a glucose molecule, or sorry, a glycogen molecule and got a glucose molecule out of it. But you may know that you could also have started at the bottom here. You could start with getting that from some other place, right? So pyruvate. You could also turn pyruvate into glucose 6-phosphate. And then you could also turn that into glucose. Okay. We want to get glucose 6-phosphate out of a glucose molecule, you use hexareverse. So lots of reversible processes here. Okay. The reason you might want to get glucose 6-phosphate out of a glucose molecule is if you have lots of glucose in your blood, right? So you're in a time period of high blood sugar, say you just ate a really sugary meal, right? Maybe you ate a donut or something. You got lots of glucose. You don't need it necessarily right now this second, especially if you ate it like right before class or something, or you eat a donut and then take a nap, you're having a nice lazy Sunday, right? So you can take that glucose, you can use our enzyme hexokinase, turn it into glucose 6-phosphate, and then do glycogenesis, making glycogen, turn it into glycogen, which you then store for later. So you can see that there's a lot of complexity in this system, right? Glycogen is our storage molecule for glucose. We need enzymes in order to break it down appropriately. They're only present in specific tissues, most importantly the liver. We can also build up glycogen when we have lots of glucose but don't have high energy needs. We don't need to know this on a super detailed level at this point, but when we get into our section on the endocrine system and start to talk about glucose regulation and insulin and glands, this stuff is going to come back. So that's why we're mentioning these words now. And that's when we'll go in detail about the gluconeogenesis and the different tissue types and who can do what and when. So this is a kind of save for later sort of topic. Right now, we just want to know glycogen is our storage molecule. We can break it down. We need that enzyme to do it. Now, most of your energy that you're storing is not in the form of glycogen. So the glycogen is just in those two specific places. In order to store energy throughout your body in big quantities, you're going to store it as a triglyceride. Remember, a triglyceride, the lipid, so it had a glycerol backbone, and then it had 3 fatty acid chains. That's why it's tri. So tri for 3, glyceride for that glycerol backbone. It's kind of like a flag or something. Okay. So it's made up of those 3 parts. The glycerol backbone is this top part I've drawn. Fatty acids are those squiggles. Okay. Storing your energy in the form of a triglyceride is really helpful because you can cram more energy into those bonds, essentially. So all of these corners, all these points are carbons. We know we just have carbon, hydrogen all over the place in our lipids. And that forms a really sort of dense energy storage mechanism. And we'll look at kind of how in a second. So they have more energy per weight than carbs or proteins. And we happen to store them in a specific cell type called an adipocyte. So that's a fat cell, the adipocyte. So site at the end tells you it's a cell type, that's for cell. Adipose tissue would be like fat tissue. But the specific molecule is the triglyceride. So when we're metabolizing fats, what we're talking about is metabolizing those triglycerides. We're starting with a triglyceride. And the first thing we're going to do is we're going to break off those 3 fatty acid chains from the glycerol backbone because we're going to treat them differently. Okay, so there are 3 of the fatty acids, there was one glycerol backbone. Let's take a look at that glycerol first. Okay. So for that glycerol, that backbone, we're going to essentially just feed it into our aerobic cellular respiration. So we're going to send that glycerol essentially into a sort of slightly modified glycolysis process. And then it's just going to go through as usual. So kind of just how glucose would. It's a little less effective at producing ATP than glucose is. So we get about 20 ATP there instead of like 30. Okay, so slightly less effective, but still decent. Where we really get the density of energy in our triglycerides is from those fatty acids. So our fatty acids are broken down through a process called beta oxidation. Okay. So what's happening here is that instead of going into glycolysis, we're pretty much taking ourselves directly into the Krebs cycle or the TCA cycle and still doing oxidative phosphorylation, all that electron transport chain stuff at the end. The reason though that this is so effective is that a fatty acid chain, actually going to write stuff out here for a second. Okay, so right, we have carbon, 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 carbon at all our points. Okay. And then we have our hydrogens coming off. So hydrogens all around, right? So I'm drawing us a saturated fatty acid, hydrogens everywhere we need to fill in the 4 bonds that carbon forms. So over and over and over again. So what's going to happen is that we don't take one fatty acid and just put the whole thing into the Krebs cycle. We're going to chop that fatty acid into chunks, and each chunk gets to go in on its own. So when we're looking at this fatty acid, and this one I've drawn is really short, we saw that when we were looking at our diagrams of triglycerides, those fatty acid chains went on for a while. So what we're going to do is chop them in groups of 2 carbons, basically like that. Right? So here, got like 3.5 on this one I happened to draw. Should have made it an even number. Okay? So every 2 carbons with their associated hydrogens is independently going to get turned into acetyl-CoA. So it's going to go through the Krebs cycle and through oxidative phosphorylation. Okay. So these 2 individual carbons and hydrogens only make us 17 ATP compared to the like 30 that a glucose molecule did. But if you look at the whole fatty acid chain, we've got 2, 4, 6, 8, 10, 12, et cetera, et cetera, et cetera, in terms of how many of these little carbon-carbon chunks we have. So as you start to multiply that 17 per fatty acid chain, right? So here we just multiply it by 3, I guess, right? But this is a really short one because drawing takes a little bit of time. Right? You can see how this rapidly stacks up. So if you have a fatty acid chain, you end up being able to produce a lot of ATP because you have all that energy stored just in a sort of tiny area in terms of how much space you have to take up for that carbon-to-carbon bond. Okay. So that's beta oxidation. We're taking just 2 carbons from our fatty acid chain. We break the whole thing up. Every 2 carbon becomes an acetyl-CoA plus some electron carriers. The acetyl-CoA goes into the Krebs cycle. The electron transport chain does oxidative phosphorylation as usual. Only produces 17 ATP for each 2 carbons, but if we think of a whole triglyceride molecule, we're going to be adding 20 ATP from the glycerol, 17 for every 2 carbons in all 3 of those fatty acid chains, and you can see how that really rapidly builds. So that's why we like to store our energy in fats. Super, super effective, takes up less space than struggling with something else. Finally, we have our proteins. Okay, so this is our last resort. So in proteins, we can do something called proteolysis. We're going to be less specific here because amino acids differ from each other. And we have 20 different kinds. But essentially, you're going to take your protein, you're going to chop it up into its amino acids. So we remember that we have a central carbon, we have a carboxyl group, we have an amino group, and we have an R group that's variable. Okay. So we're going to first deaminate. So we're going to remove the amino group. Right. So that's what happened here. We took the amino group off. Okay. It's going to leave us with keto acids. Okay. Our keto acids are going to become essentially pyruvate or acetyl-CoA, and they're going to go into the Krebs cycle. This isn't a great storage mechanism in terms of using energy, though, because our proteins are mostly in the form of muscle, and we want to keep our muscles. Or enzymes, for example. You don't want to break down an enzyme for energy. You're using the proteins to do stuff. Okay. That brings us to membranes. We're going to start in a new package. So as we're waiting to pull, maybe we don't have that many people using physical copies these days. Hopefully this is reviewed from your previous courses. We're going to go through what a scientific kind of. So the big point of this is that we want to understand how cells work. So part of understanding how cells work, we need to know what can get inside under what conditions, what leaves under what conditions. And in order to figure that out for different tissue types, different parts of your body, we need to know what a membrane is. So we're going to first talk about how we build a cell membrane, and then we're going to move into thinking about the forces that push molecules across the membrane. So when we're thinking about the plasma membrane, usually referred to the picture would come up here, or if you drew one, you can draw one. This is a slide you should draw on. We use something called the fluid mosaic model. So the fluid mosaic model is referring to the fact that cell membranes are kind of jiggly. We don't have cell walls, we're not plants. Our cell membranes can move a bit. They can expand, contract, bud off little pieces, fluid. And it's a mosaic. So it's a number of different types of biomolecules, of different molecules that are making up this larger cell membrane. So we think about it like a mosaic on a wall or made up of many different types of tiles. So our different pieces that we're thinking about, our mosaic is going to be made up of phospholipids. So hopefully you remember that those were our amphipathic group of lipids with its polar phosphate head, its non-polar fatty acid tails, giving one side hydrophilic or water attracting properties, the other side hydrophobic water repelling properties. We also have cholesterol, which is also a lipid. So cholesterol was the basis for our steroids. So we saw cholesterol was like 4 little rings kind of pressed right up against each other. So it's still a lipid because it's mostly made up of carbon and hydrogen. We also have membrane proteins. Membrane proteins are going to form a lot of important structures in the cell. They're going to help cells stick to each other. They're going to give the cytoskeleton kind of anchor points. We won't talk about those two pieces much in this class, but we will talk a lot about receptors. So they're going to get signals from other cells. We'll talk about transport proteins, so things that can move stuff across cells. Finally, we have some membrane carbohydrates, which are essentially just going to be like little kind of stickers stuck onto some of these other molecules. So we're going to start with those phospholipids because they are the basis for the structure. So the phospholipids make up most of the cell membrane. We saw that phospholipids kind of naturally orient themselves in water in specific ordered structures because we have one side that's polar and likes water. We have one side that's non-polar and hates water. Okay, so here we have our phospholipid bilayer. So here's one phospholipid. Here's another phospholipid. They're kind of lying foot to foot or tail to tail in the center here, with those polar heads touching extracellular fluid, or ECF. Okay, so that was one of our types of fluids. And touching our intracellular fluid in the cytoplasm, so our ICF. Cool. So polar heads, those circles touch both parts, fatty acid tails in the middle. Okay. So the phospholipids form a really difficult barrier for water-soluble substances. The water is a polar molecule. So H2O, that O, that oxygen, highly electronegative compared to hydrogen. It kind of hogs electrons, creating these subtle little charges on the molecule. Okay? So this means the things that dissolve in water, things that mix up well with it, break apart in water, are going to be things like ions, right? So ionically bound compounds break up, they each get their own charge. They're either going to be attracted to that charge on the oxygen or repelled away from it. But either way, that substance is breaking up in the water. Right? So that's what makes something water soluble. So when we say a water soluble substance, we're thinking of something that kind of has a charge somewhere on it a little bit. Okay. So why is this creating a barrier? If you'll notice, looking at the thickness of this membrane, right, it is true that we have a polar head touching the extracellular fluid and the intracellular fluid, but most of the thickness here is made up of those fatty acid tails. So those polar heads are attracted to the water and the fluid on either side of the membrane. But if you're thinking about a molecule that has to get across, right? Very little of it is spending time trying to pass that polar head, but it would have to spend a long time crossing this region of fatty acid tails before it finally gets into the cell. The fatty acid tails are what makes it a lipid. They're non-polar, they're hydrophobic. They're hydrophobic because they're repelling anything that has a charge. So something that's water soluble probably has a charge somewhere on it, which means those fatty acid tails in the center of our membrane are going to repel those things just like they would repel them. Okay? So this sort of uncharged region, this non-polar region in the center of the membrane, really hard for anything to cross that has a charge. So specifically what I'm saying to you now, our application bit, our ion that we'll talk the most about, like forever and ever in this course, is sodium. So sodium comes from sodium chloride, salt, right? So NaCl, right? Breaks up, gives us Na plus Cl minus. Sodium drives all sorts of things in the cell, but because it has this charge, it can't cross through the fatty acid layer here. It can't cross through this hydrophobic layer. So anytime we see sodium crossing a cell, we're going to need to make a door for it. Okay? That's because of this non-polar region in our body. Other things that the phospholipids are doing, providing some fluidity. So these sort of stacked phospholipids right next to each other, those stacked fatty acid tails, that's part of what makes it jiggly. Okay? Technically, our phospholipids are, well, for sure, our phospholipids are impermeable to water. That hydrophobic region, that non-polar region, those fatty acid tails, they're repelling water. But when we talk about cells, until we get to the kidneys, we are going to be just assuming water with them. That's because, well, water can't go through the phospholipid part. Water can go through a specific type of protein called an aquaporin, right? And almost all of our cells, we're just going to assume that they have aquaporins. aquaporins. Aquaporins are super common throughout cells. So unless we specifically say that we're talking about a cell that has no proteins, that has no aquaporins, we are going to assume that water can cross because the aquaporins are there. That's because of the proteins. The phospholipids themselves would repel water. Okay. So our third piece of the membrane is our cholesterol. So the cholesterol is also contributing to the fluidity of the membrane. So it's interfering a little bit with the interactions between the phospholipids. Under certain conditions, if we had just phospholipids, they may crystallize, like just kind of get stuck in place like an ice crystal, but the cholesterol molecules kind of jam them out of the way and mess up that process, which is good. And it can also interfere a little bit between interactions there. We're not going to talk all that much about the cholesterols in the membranes in this course, but you should know that they are there. Another thing that they do is the ability of the membrane. The proteins are super important to know about. If I'm ranking components of our membrane to understand, you want to understand the phospholipids and you want to understand the proteins first. The other bits are kind of extras. Okay. They're there, we should know they're there. For future courses, you may talk about them. But for us, phospholipids and proteins. Okay. So if we are looking at a protein, these purple things all throughout here, okay, we see that they are in some way either embedded in the membrane or they're just kind of sitting on top of it, either on the inside or the outside. So purple everywhere here is proteins. So if you wanted to isolate one like in a lab and study it, you'd have to disrupt the membrane and just kind of like pull it. We're not doing that in this class, so don't worry about that too much. The ones that go through, all the way through the membrane though, do need to be able to embed themselves both among those polar heads, those non-polar fatty acid tails, and again, same deal on this side, and they need to be able to touch the water in the extracellular fluid and the water in the intracellular fluid or in the cytosol. So if you see a membrane protein that goes all the way through, or that embeds itself even partially in the membrane, it's going to have that same property that the phospholipids themselves have, which is that part of it is going to be hydrophobic. The end of this word. Going to have a hydrophobic part touching the fatty acids here. It's going to have a hydrophilic part, a water-loving part, touching the ECF and the ICF, as well as integrating with those polar heads of the phospholipids. Okay. So, some words, specific words. An integral membrane protein means it's integrated into the membrane. So that's one that's going at least partway through. Okay, so touching both parts. We refer to something as a transmembrane protein, trans across membrane for the membrane. So a transmembrane protein goes all the way across. So a transmembrane protein would be a type of integral membrane protein. Okay. And we'll also have peripheral membrane proteins. Those just kind of sit on top. So they wouldn't need to have these same amorphic properties necessarily. The most important proteins in the membrane that we'll talk about for this unit, and the proteins that will be critically important throughout the rest of this course, are going to be channel proteins and carrier proteins. So those are going to be proteins that go all the way across. So they're going to be integral proteins, they're going to be transmembrane proteins, all the way across, embedded inside. Those are going to be our channels and our carriers or transporters. So these are going to be the ones that allow things to go through the membrane. So channel will either have literally like a channel inside it, like a space. Okay, so a channel protein. be literally a channel where something could go through. Carrier proteins will have to move. If I move, I need a carrier protein, will have like a pocket on this side, but then sometimes splits to the other side. We'll talk about those as we talk about transport across membranes and as we talk about systems where this is an important part. The other proteins are also doing important stuff. We'll just think about them less. So we will think about some of the proteins that work as enzymes. Becomes part of it in digestion. We'll talk about receptors receiving signals as well. But the first thing we want to understand are transmembrane proteins that act as channels and carriers. Peripheral membrane proteins are the ones that are just kind of sitting on one side. They can pop up pretty easily because they're not embedded in the membrane, they're just sitting on top of it. Most of them happen to be touching the cytosol, so the intracellular fluid, rather than the extracellular fluid. And most of them are like anchor points for parts of the cytoskeleton, which we won't talk about in detail in this course. Our final group are our membrane carbohydrates. They are bound on to either lipids or proteins. So we can see both here in this diagram. So these branching are the carbohydrates that we're talking about. So when you're looking at that branching, think kind of like the glycogen had branches where it was holding together its glucose monomers. Right, that's what this branching is that was happening here. So this one is holding on to a protein, so that makes it a glycoprotein. Okay. The glyco is telling you that this has a carbohydrate piece. Protein tells you it's bound to a protein. We also have glycolipids. So here you see one's just stuck onto a phospholipid. Okay. So a glycolipid is a carbohydrate stuck onto a lipid. I think we'll mention this at all again in this course, but these do function in cell recognition. So how your cells know what other cells are around them, whether you have foreign cells or something. And in other courses, you'll talk about how they're part of like the glycocalyns. You can presumably talk about it in like microbiology. All right. So our functions for our membranes. The membranes are going to allow for transport. When we think about transport, trans means across. So transport, we're talking about how things get across membranes. Membranes get to choose what can cross for the most part. Okay. So when we're thinking about a membrane, we're going to draw our typical cell, just a typical circle, right? So the first thing to realize here is that we're separating two of our types of fluid. So our intracellular fluid, our ICF, is on the inside. Our extracellular fluid, our ECF, is on the outside. Okay. Said our ECF is essentially our internal environment of the body, so the external environment for a cell. Okay. So we're separating this material, but we are going to want things to be able to cross between these two types of fluid. We're controlling what and when, but it would do us no good if we can never get anything that's in a cell out, and we can never get anything that's in, for example, the plasma in our bloodstream, which is one of the types of ECF, into a cell. Okay. So it's going to be important that we can let stuff cross. For example, we're going to want to let O2, so our oxygen, in so that we can actually do metabolism. We're going to want to let things like glucose in, right? Because we want the sugars to break down to make ATP. We're going to want to get rid of waste products. One of our big waste products is going to be CO2. That was a byproduct of breaking down glucose, right? I'm also choosing these because it's really obvious, because we breathe in O2, we breathe out CO2. This is pretty hopefully easy to realize that these things must be moving around our cells. So we refer to membranes as collectively permeable. So what this means is that they are restricting what crosses and when. This has to do with that control piece. So permeable, permeate just means that something can cross. So if we say a membrane is permeable to water, we mean that water can cross that membrane. The selective piece is that because we can control what proteins are in that membrane by making new proteins or by removing proteins, we can control when, for example, a sodium ion can or can't get into a cell. So that's the selective piece. Okay. There are, however, some things that can always go through just based on their chemical properties. Those are going to be things that are small and non-polar. So they have to be small because they can squeeze in, just like shove their way through between those phospholipids. If you have something really big, that's not going to work. And they have to be non-polar because they have to cross that big hydrophobic region made up of the fatty acid tails of the phospholipids. Okay. So when we're thinking about molecules that can just diffuse across the membrane, we're going to be thinking about oxygen, carbon dioxide, and fatty acids are actually also going to be able to diffuse across. So they're small, they're non-polar. Our ions, like sodium, so Na+, and polar molecules. So polar molecules, we're shorthanding, we know something's polar, we can see kind of oxygen sticking out. So all those OH groups on carbohydrates make them a little bit polar. Water is polar. Ions and polar molecules are going to need to go through a protein in order to go in or out of a cell. They're not able to just cross that phospholipid bilayer. So unless we are allowing them through, they don't get through. Okay. So examples of polar molecules that we'll be interested in are glucose, sodium, and also proteins themselves may or may not. Or if we want to let one in or out of our cell, we're going to need to come up with a mechanism for that. So our next piece is that we are going to talk about the forces that would drive something across a membrane. So we're basically trying to predict what direction an ion or a molecule is going to go relative to our cell membrane. As we do that, we're going to need to think about our different types of transport, like passive transport and active transport. So the first piece that we want to remember when we're talking about this, we're going to set ourselves up for what we're talking about on Friday. Right? We want to remember gradients, and we want to remember our basic definitions of active and passive transport. Okay. So passive transport, what passive transport means is that this does not take energy. This is a process that happens spontaneously. It'll happen just kind of on its own. You let it go. And this is going to happen when we think about gradients. And probably what you've talked about in your earlier classes at this point is talking about concentrations of things. Right? We're thinking about it as downhill movement. So with a gradient, so we're going from areas of high, for example, high concentration, but we'll see that this is actually more complicated in real life. So high, for now, concentration, to low, for now, we're just going to think about concentration. Okay. So we call this moving down a gradient, just like rolling a ball down a hill. So that's passive transport. Active transport is going to take some form of energy for somewhere because we are rolling that ball back up the hill. So active transport is going to go up our gradient. So we're going to go from, for example, a low concentration to a region of high concentration. Have to put in some energy from somewhere to do that. This is not going to happen spontaneously. An example we've already seen is in the electron transport chain. Right, we're packing lots of hydrogen ions into that intermembrane space. We were using those highly charged electrons from our electron carriers to force them into that space, even though it was getting really crowded in it. So active transport is going to require a source of energy, usually ATP, but again, we got some other things going on as well. So leave it off there. And if you don't remember anything about chemical concentrations and chemical gradients for active and passive transport, I recommend you look that stuff over now, because we'll be expanding and detailed and modifying our understanding of that in the next